this next section, we're going to talk about microbes and their relationship to oxygen. And whenever I teach like intro principles of biology stuff, I talk about what's required for life, and we go through, 7 things that define life. And then when I have an exam, there's always about 4 people that say oxygen is required for life. Oxygen is not required for life. Oxygen is required for your life. It is not required for all life. There are a lot of microbes that not only don't need oxygen, but would be killed by it. And we're going to talk about why that is and how we classify microbes according to their oxygen use. This chart is really useful, and one of the only charts where I would encourage you to look at all the parts of it eventually. But for now, what I want to look at are these definitions over on the side. There's about 6 definitions of microbes that come out of this section, and they're listed over here. They also do have a correct description here and here, but what I like to do in this section is make a yes-no chart, that, like where you ask questions. Does this microbe do this? Yes. If so, it's this. No? Ask the next question. So we'll do that over here on the board. And those of you that are watching the video, you'll want to get this board chart from a friend in class later, because I'll turn the video off before we do that. So there are 2 big questions that we need to ask when we think about microbes and their oxygen, their relationship to oxygen. So firstly, do they use it? Do they need it like we do to support their electron transport chain? Do they use it as a electron acceptor? That's what I mean by use it. And the second question is not really related to that, but are they poisoned by it? We are not poisoned by it very much. We can deal with the toxic effects, but not all microbes can. I moved a few slides just now up in the lecture. So you've got these slides, they were just at the end, and I moved them right here. So oxygen is a great electron acceptor. Because we use it at the end of our electron transport chain, we make lots of ATP. So we get a lot of energy out of our food, so to speak, because we have oxygen at the end of the electron transport chain. It's a great electron acceptor, but it also is such a great electron grabber that it can form these reactive oxygen intermediates, or reactive oxygen species, or ROS, or these are also popularly known as free radicals. Like out in the general public, what they would be calling this concept are free radicals, which is easy to say. Same concept, free radicals. So these are the kinds of free radicals that you can get from oxygen being in a watery solution. And maybe some of these you might have talked about in Gen Chem, but the stuff in the colorful boxes is damaging because they've got that extra electron. These chemicals can take their extra unpaired electron and cause a lot of damage within cells. Free radicals, ROS, reactive oxygen species. So organisms that use oxygen have to have a way to deal with these ROS, and we do. So some of the ways to deal with these are having enzymes that do detoxification. Oxygen is a blessing and oxygen is a curse in some ways. Organisms that have enzymes like these are not the only ones, but these are the major ones. If they've got these, they can deal with the reactive oxygen species and not suffer a lot of cellular damage. So catalase, peroxidase, superoxide dismutase, take aparter, dismutase, superoxide reductase, these are protective enzymes. These are protective enzymes. And any organism that uses oxygen would have to have at least one of these. And they usually have more than one of these at a time. This one particularly, catalase. Do you remember that equation? You ever seen that equation before? That is the chemical equation that you need to know in this class, specifically for lab. We're going to be doing catalase tests in lab. So when you're looking at this catalase part up here, catalase is the arrow. Catalase is the enzyme that does that reaction. So it takes two hydrogen peroxides, which are ROS and damaging, and it breaks it down to water and oxygen, which are not particularly damaging. That's what it does. So pretty simple equation. It's catalyzed by an enzyme. And we use this process in lab to determine whether a microbe has catalase activity or not. And it's a really nice little reaction. This is called catalase test, which is on there. So when you do a catalase test, you're trying to determine whether your microbe that you're looking at has catalase enzyme. And what you're looking for as a sign of that is oxygen bubbles. So when you see this side, there's this drop over here, comma, peroxide dropped onto it. So now that's this side. And if you drop peroxide onto a microbial solution and you see oxygen bubbles coming out of it, that's an indicator that it does have catalase. If it just sits there and it's like this and you don't see bubbles, that's an indicator that it does not have catalase. So we say this would be catalase positive result, this is catalase negative.
So only the ones that use oxygen have these enzymes?
Organisms that can tolerate
So
oxygen.
the ones that even tolerate oxygen?
They have to have one of those enzymes, yeah. We'll get into that in more sophisticated detail in a minute. Yeah. If they use oxygen, they must have these. If they just tolerate oxygen, they also must have some of these.
Okay.
Yeah. I want to jump ahead a little bit to think about, all right, if there are microbes that don't use oxygen and can't tolerate it, how did they even end up on Earth in the first place? Why should you be on Earth if you can't tolerate oxygen when it's, 19% of our air?
Well, if you're deep in the soil.
If you're deep in the soil, that can be a place where anaerobes, which are the ones that don't use oxygen, anaerobes would live. Deep in the blank is a good place to think about. That's where anaerobes live. Deep under the ocean, deep in the mud, anaerobes. So we'll get into this in Unit 3. But the main reason why we have anaerobes on Earth is that anaerobes as a group, they're older than oxygen. Earth didn't always have oxygen. So the ones that don't use it, they're very ancient in their lineage. We talked about that in plant a little bit, right?
Yes.
Yeah, we're going to talk about it again later in this class. But oxygen wasn't always on Earth, so oxygen's newer than these ancient anaerobes. We'll come back to that later in class. But if you wonder, like, how could there ever end up being organisms that don't use oxygen?
I have a question.
Mm-hmm.
Why would an anaerobe want to live in the upper respiratory tract? I know it's aerotolerant, so it can be okay with oxygen, but why would it want to be in an area where there's constantly oxygen flowing in?
Because for that microbe, so the question is why would an anaerobe ever want to be in the upper respiratory tract when there's lots of oxygen? It has to be aerotolerant to do that. It has to be able to detoxify.
Mm-hmm.
It's not there for the oxygen levels. It's there for a food source. So, what brought you here today? Food. I'll deal with the oxygen. But, notice what that is, Strep pyogenes. It grows, if it could have food and not oxygen, it grows better. And it actually grows better under the surface. It grows better under the surface of your cells than out on the top.
Okay.
So it prefers to grow deep where oxygen is less, which is kind of gross, but makes sense. And when you grow it, when you grow it in agar, it'll grow better if you stab it down into the agar, and it's just a little bit shielded from oxygen under the top. It's awesome. It's a good question though.
Does it require a lot of energy with their enzymes to fight against like oxygen?
Does it take a lot of energy for the enzymes to detoxify
Yeah.
oxygen?
Is that why they do better?
Notice in this category it's saying it doesn't require oxygen. It doesn't grow better when there is oxygen. If it took a lot of energy to deal with oxygen, that's probably measurable if you set up an experiment really well, but it would not, if it finds a great food source in your throat, it'll make up that deficit, if there is a deficit. There probably is a deficit, but it's probably so small that it's not too much involved in the
Where-
decision, so to speak, of where to colonize.
Yeah. Okay.
So, what I want to do next is actually do the chart. Don't necessarily need to have that on video, but I want to talk about this slide, these two slides, and then we're going to do the chart, and I'm going to come back to this slide and use the information to answer this question. So, taking those definitions that are on the chart and that we're going to go through off camera, classify what kind of microbes are in each tube. This kind of picture is really common in all microclasses, and it takes a little bit of setup to understand what this is. So these are specialized broths that have an oxygen gradient top to bottom. I think they're called thioglycolate or something like that. Specialized broths, and as you get closer to the bottom of the tube, there's less oxygen. And up at the top of the tube, that's at the room level of oxygen. So this up here, in the air in the tube, same air that we're breathing, 19% oxygen, the oxygen will make it down a little ways into the tube, and then it becomes less as you go down. It's generally not like such a sharp cutoff as they're showing in this cartoon, but generally as you go down there's less oxygen. So what you're looking for when you look at the tubes is where are the microbes growing. On the cartoon it's easy. On a live picture you're looking for cloudiness. So if you inoculated these tubes, you would want to get them out carefully out of the incubator. Do not shake them. Do not mix them. Look at, are the microbes growing at the top only? Are they growing at the bottom only? Do they seem to have no preference about anything? They're just growing evenly throughout? Do they grow slightly under the top? Do they grow throughout but a little bit more at the top? That's what you're looking for. So where does the microbe grow? What is the oxygen level like there? And then you can use that to classify what kind of microbe it is. The last slide in this section is just to show that if you're going to work with anaerobes, you have to do something about this 19% oxygen in our air. You can use these gas pack materials, which is basically taking a catalyst that removes oxygen, break the seal, put it in a sealed bag. It chemically reacts the oxygen to take it, to convert it to something else, and then this becomes anaerobic. That's a thing that we could do in lab here. We may actually try it in a few weeks. I've got some really old ones of these. We'll see if they still work. If you're going to seriously do work with anaerobes, you have to do it in a glove box, which is tedious. So this is a sealed chamber. You've got glove holes to put your hands in. You do all the work. We're actually going to go to Eastman. We're going to go to Eastman Microlabs later, and they've got one of these. Did you go already? You don't have to go if you don't want to, but it's going to be a slightly different trip. They have one of these that they use kind of regularly that we can look at. So. Okay, if you'll hit the button, if you're watching the video, you'll want to get this chart later from a friend.